Um, yeah, it's a pleasure. So thanks. And what I'm going to talk about today is some of the work that my lab works on, and I'm going to include some earlier work that I did from my postdoc and stuff that was done with traditional tools, and then I'll tell you about some cutting edge tools that we're starting to apply to bacteria. And really, as the title shows, what my lab works on is in single cell analysis. And we try to look at how heterogeneous a cellular population is. So how many different cell states are there in a bacterial population? And I'll go into these pictures in a second. Oh. But really, I wanted to start actually with mammalian systems because that's where we're more familiar with thinking about multicellularity. So when we think about the human body, for instance, we know that there are over 200 cell types in the human body, and that each of these different cell types have a different function. So we have bones to give our body structure, and we have muscles so that we can move around and do things, right? We have all kinds of immune cells to protect us. And we have a brain that can control all of this. And so when we talk about multicellular behavior in a multicellular organism like a mammalian system or a human, it's very easy to understand. But when we actually talk about it in bacterial communities, there's actually also a lot of complexity and multicellular interactions in these systems. And so typically when we think about multi-species bacterial communities, we think about the human gut microbiome or about some infectious diseases, which are polymicrobial, so multiple species work there. There are some foods in which you use multiple different types of bacteria that work together, soil and aquatic communities. But I'm not going to talk about that today as much. What I'm really going to talk about is a single species bacterial community and whether we actually have multicellularity when bacteria grow in isolation. So a pure culture. And what I'm going to try to convince you is that bacteria can form specialized cell types even when they're grown alone, and that these specialized cell types are important in disease, industry, and the environment. Okay, and so let's talk about this a little bit because I think that that's not usually the way we think about bacteria. But even from the beginning of microscopy, people saw different cell types. So this is a picture from the Botanical Gazette in 1892. A picture was taken by Roland Thaxter, who at the time was a professor of botany in Harvard University. And this picture, even though he was a botanist, these pictures are not plants. They look a lot like plants, but these are actually bacteria. And these are a mix of bacteria. And you can see that on the top of these branches that look like tree trunks, there are spores, right? And so you can argue that we've known that there are different cell types for the longest time. We've seen it microscopically, but we've not been able to really study it, right? And so this goes beyond beautiful things like myxobacteria. We also see this in other organisms. This is a picture of Bacillus subtilis. And in Bacillus subtilis, we actually see multiple cell types. You see spores just like the myxobacteria, except that here they don't form those beautiful structures. We also see these cells that are red. These cells are competent cells. And the only reason we can see them is because we made a fluorescent reporter that basically puts the promoter for this gene driving a fluorescent protein. And so the red here are basically cells that turn on competence. And so they're able to take up DNA from the surrounding. And so I started talking about the human body and about antibiotics. But in reality, when we think about the human body, the reason we have these different cell types is so that we can do things that any cell cannot do by itself. In bacteria, the way most people think about these different cell types is that the bacteria are basically hedging their bets. The bacteria are sort of betting. They're saying, oh, maybe the conditions are going to get super, super bad, so we should have some spores. So if conditions get so bad, some of us will survive, and then we'll be able to basically grow out. Or maybe the conditions are going to get pretty bad, but there's going to be a gene in the environment, and we'll have some cells that are competent, and we'll be able to take that. And so microbiologists call this bet hedging. The bacteria are basically betting on so this is very different than in the human. It's not for immediate, in real time reasons. It's for some future results. And when I started my postdoc, I thought, wait a second, is that really all it is, or is it other stuff? And I thought maybe we can look in metabolism. And so here's a picture of metabolism. You don't need to know all of this, but in metabolism, do we actually see heterogeneity? Because then it's probably something that the bacteria are doing for immediate use. And when you look at these maps that we are used to looking at, they're actually a little bit misleading, because the map is sort of like a traffic map in a big city, but it shows you a static representation. It doesn't show you if the stoplight is on, if there's a red light. It really doesn't show you which cell types are active in a particular pathway. It doesn't show you how metabolism is organized in space, maybe different bacteria in different parts of a colony. And it doesn't tell you what actually might regulate this structure. And so what we did is we basically looked at central carbon metabolism in single cells, and we made fluorescent reporters to different genes in the TCA cycle or in glycolysis. And we see that even in the TCA cycle, there are some genes that are only turned on by some cells in the population. And then when they go out of later stages, there are other genes that are turned on. And we can take these images. You might have seen that this is actually an image of the same field of cells. It's the same cells with two reporters. And so it's not just one or two genes. It's genes that are maybe correlated or anti-correlated. So you can see here that cells are either red or green. So they're either doing the TCA cycle or making pyruvate. And we can now look at some of these dynamics. So when we look at this, we can, since we have these fluorescent reporters, we can put bacteria in growth chambers and flow media on top of them and see how often do these cells switch. So these are cells that are switching into that condition that I showed you, which is a TCA cycle gene. And you can see that cells that actually enter this, like these cells over here, they grow very slowly. And they'll come out of that state, but they grow really, really slowly for a while. So that's very different than the way we think bacteria grow, right? We usually think all the bacteria grow similarly. This is the other gene that I showed you, which is a gene later on in growth. And you can see that these cells switch in and out much quicker. So the next thing we wanted to ask is, unlike bet hedging, is there maybe some reason? Oh, so with these pictures, we can actually just display it in a static picture. We can take the same well over time, and we can show you two sister cells, and one of them grows slowly and one grows fast, and we can measure the switching rates and the dynamics and how fast these cells grow and divide. So we wanted to see whether the metabolites themselves are actually influencing this. And the way we did this is we took snapshots of cells, and we counted how many cells are in the off position and how many are in the on position. And we knew that acetate is driving some of this differentiation, because some of these cells are responding to metabolites. We see that when there's no acetate in the media, basically all the cells, or almost all the cells, are off. And as we add more and more acetate, more and more cells turn on. But interestingly, some cells are off even when you have a lot of acetate. This is very different than the way we usually think about how cells regulate their transcription. Usually we think that if there is more stress, every cell is going to make more of a protective protein, right? Here I'm saying it's not that every cell makes more, it's that more cells are entering that state. And so this is very, very different. When we grow the cells in a different type of movie, where now they can actually experience their neighbors, we see that cells are going to turn on one of these metabolic genes, which we believe is for detoxification of short-chain fatty acids. And some cells don't. And I'll show this again with the lineage tracing. You can see that the cells that turn it on are going to turn it on right about now. And all the cells grow slowly at this stage. But after they turn on for a while and detoxify, you can see some cells start to grow really fast. I don't know if that's clear to see, but you can see it also in the lineage trace. Cells that are on have a dark red or yellow promoter activity. They divide a few times, but cells later on in the movie basically divide much more quickly. And what we think is happening is that cells are basically responding to their neighbors and are able to detoxify their environment. And so bacteria really start to influence each other. And so the picture that we have is that we basically have cells that produce acetate. We found one gene that was a marker that we made fluorescent reporters, and then we basically were able to find the entire regulatory system and confirm that these cells make acetate. As the acetate increases in the environment, these detoxifying cells start to arise. And as they arise, they basically counteract the acetate by making neutral metabolites. And so basically, and the acetate gets reduced, and then cells can grow faster. So in this first part of the talk, hopefully what I've been able to show you a little bit of is that in clonal bacterial cultures, cells can actually perform specialized metabolic roles. The secreted metabolites themselves can help cells actually control some of this specialization. So here we saw acetate turning some cells on and off. And the last thing that I showed you is that these specialist groups can actually work collectively and benefit the entire community. So a little bit like the analogy that I had with our cells of our body that work together. You know, I need to drink water, so my arm is going to grab my cup and I'll drink. The same thing here, the bacteria are working together to benefit the entire organism. Okay, so now I wanted to switch into what my lab is doing currently, because this is sort of the reasons why we're really interested in heterogeneity and we want to understand it. But I showed you this picture earlier, and I was very proud that we can actually see two cell states in one picture. But in reality, the question is, how many cell states are there in this picture? There are cells that are not green or red, like these cells down on the bottom. There's cells that look sort of fat and cells that look sort of skinny. How about the cells that are green? Maybe these are like T cells. Maybe there's T helper cells and T killer cells and B cells, right? So the fact that we can see two colors is wonderful, but is that really all the heterogeneity? And maybe there is 50 different cell types in here. How do we actually know? And so I really wanted to start understanding this and to try to look at the entire transcriptome of cells, and not just by making 1,000 reporters, because you cannot look at 1,000 colors at the same time, but by doing single cell RNA sequencing. And in mammalian and other eukaryotic cells, people have been doing it for the last few years using microfluidics. And my lab decided that we want to sort of create a system for bacterial single cell RNA sequencing. And so the way that these systems for mammalian cells work is the most common ones is you have basically hydrogels that come with about 1 million different flavors. And what makes each one of these hydrogels, the green and the blue and the orange, different is they basically all have a primer on them with a unique sequence that is different for each colored barcode. So the green one will have one sequence and the blue one will have another one. And then in the end, there's a poly T. And basically through microfluidic encapsulation, you can encapsulate one of these droplets with a single mammalian cell. And because these are mammalian cells, these T's in the end of those single-stranded DNAs can now bind to poly A's on the end of transcripts. And if you add reverse transcriptase, what you're going to do is you're basically going to add this cell barcode to this mRNA. And then if you sequence these, you can take all the cell barcodes that are green, meaning there was the green cell in there, and read all the mRNAs that cell basically encoded. And so that is basically what is done in mammalian cells. This is what it looks like in a microfluidic device. These are those beads that I was talking about. This is slowed down about 30 times. It's a very fast process. There's going to be cells that are flowed on one chamber. They're reverse transcriptase on another one. Those are the beads. And as they come across an oil junction, you can see that there's one of those beads in each droplet. And you can see that most of these droplets only have the bead. They don't have any mammalian cells. There's going to be one that comes now with a blue dot arrow. You see there's a cell that came with it. But they basically will have, most of them will have no cells. That's how we make sure that it's single cell. There's some that have no bead, like this one actually had a cell in it. And there's another one with a bead and a cell. So that's how the system works. So why is this so hard to do with bacteria, right? Well, bacteria are very different than mammalian cells, and so there are a few challenges. The first and most important challenge is there's no poly-A in bacterial cells. I mean, there's very little of it, and so we cannot use it. Also, the mRNA content is much lower. There's only about 1,000 transcripts in a bacterial cell. There's maybe 100,000 in a mammalian cell. The mRNA half-life in mammalian cells can be 10 hours. In bacteria, your mRNA will degrade in 2 minutes or 5 minutes. And so you have very short half-life. You have to do something about that. The ribosomal RNA is about 95% of all your transcripts is ribosomes. And so if you just try to sequence, all you're going to get back is a ribosome. And the final thing is that these bacterial cells have a cell wall. And so within that drop, the next thing that happens is that the cell has to lyse so that you can actually do your molecular biology on it. Well, if you have a really thick cell wall, it's going to be very hard to lyse those cells. And so what my lab basically decided to do is we're going to use the same pre-existing microfluidics that I just showed you, but we're going to retrofit the bacterial transcripts. We're going to change them so that they actually have a poly-A. And we're going to do this by making very large-scale oligonucleotide probe synthesis. And we're going to start by using Bacillus subtilis as a model organism, just because it's a really fun bacteria to work with. And so the reason we thought this would work is because people do fish with fluorescence in situ probes against the transcriptome, and you can actually count dots in bacteria. Here it's a yeast, but you can do it in bacteria too. Each one of these dots actually represents one transcript. And so they can do it with fluorophores. The problem is they can only count up to maybe 5 genes because they only have 5 fluorophores. We thought, hey, maybe if we put a poly-A tail on the end of our probes, we can actually encapsulate them. So this is what this actually looks like in a schematic. So the first thing that we do is we take our bacterial cells and we fix them in formaldehyde. So they're fixed. So now we're not worried about the half-life of the mRNA. The second thing is we permeabilize them. We add an enzyme that basically degrades the cell wall. So now we're not worried about the cell wall. Then we add these probes that target the mRNAs. And the probes have a poly-A on one end, and they have a PCR handle on the other. And they have one region that basically is complementary to the mRNA. And so that will bind. Now, our probe set has about 30,000 different probes. And it's actually cheaper to make than you would think. It's not cheap, but it's cheaper than you might think because you can order them from a microarray and then just amplify them up. And so it costs about the same to order them the first time as it used to cost to get a microarray. And then after we bind them, we wash off all the probes that are not bound. And then we basically encapsulate them in droplets just like we did before. And when we do this for Bacillus, we did it for just one time point, right? So we take Bacillus, we grow it in a very well-shaped condition in minimal media in late exponential phase. So it's not even in stationary phase. And this is what we see. We basically see that even in a well-mixed environment, there are many different cell types that exist in bacterial population. So if you look at these genes, I didn't arrange them, but each one of these columns is a cell. And the rows are marker genes that the program chooses as genes that are differentially expressed. And so there's about 3,000 genes in this picture. And the program basically arranged these cells by how similar they are to each other. What you'll see is that a lot of these genes, this big block of overexpressed genes, so overexpression is in yellow, and if they underexpress something, is in purple. So all these overexpressed genes here all start with COM, or many of them do. This is competence. Remember, I showed you earlier that we already know that genetic competence is only in some cells. So we can actually see that. But instead of just using one reporter, we can see all the genes that come up in competence in these cells. The other thing that we see here are these genes that have SPO. That's sporulation. So I showed you, when I showed you competence, I showed you sporulation as well. But there are other groups here that are not sporulation. And so if you look at this cluster, these are all genes that start with ARG, A-R-G. That's arginine metabolism. We actually recover the entire operon. Actually, a couple operons for arginine synthesis are only expressed or differentially expressed in a subset of cells, right? And after we did this, we went back and saw how much of the operons we see. So for competence, we see 90% of all the competence genes, and we can make fluorescent reporters, and we can see that the percentage of the population that we saw is the same that you see with the fluorescent reporter. You can do the same thing for sporulation, and you can see a very nice phase bright spore. And we have a GFP in a spore coat gene. And so we can actually see a whole bunch of sporulation genes that are upregulated in this population over here. And I mentioned arginine, so we can actually see that with fluorescent reporters, not as strong, but look, that's the entire arginine biosynthesis pathways upregulated in these cells. And so I think I'm scheduled to talk for about 30 minutes. So the last thing I wanted to talk about is why do I think bacteria do this? And
no, Adam,
arginine,
you have sorry, Adam, you have 1 hour. No, don't have hour. For you is 1 hour. No worries about that. Sorry.
I scheduled for a half an hour, but we can talk more afterwards. I have other stuff that we can talk. So I've been rushing a little bit. I took some slides out, so hopefully, but what I wanted to talk about is why I think that bacteria are really doing this. In arginine synthesis, we've done this now for several bacteria, not just for Bacillus. We've done it for about 5 or 6 different bacteria, and we see arginine synthesis in a subpopulation of cells over and over. And what's really interesting is that arginine synthesis is also differentially expressed in both plants and mammals. So in animals, and mammals in particular, it's expressed in different organs. So in the kidney and the liver, and even in these organs, there are different parts of it that happen within different organelles within cells. Within plants, part of it happens in the mitochondria, part of it happens in the chloroplast. Okay, so basically, this particular metabolic pathway is segregated into some locations or some cell types in all of these systems. And what I think is interesting about that is I think that what it means is the reason that we need to have multicellularity, or one of the reasons, is that maybe some metabolic reactions, some biochemical reactions might not be compatible with each other. Maybe you cannot do arginine metabolism if you're making tryptophan, or I don't know, if you're doing something else. I don't know what it is yet because I haven't found it, but that's sort of the idea. And I just sort of wanted to share this example of a really interesting bacteria in which this exact thing actually happens and is known. And so I started the talk by saying that we don't really fully understand how bacteria in real time interact. And that wasn't entirely true, because there are a couple of examples, well, maybe one example, where there are bacterial interactions that benefit the population in real time. And that example is with Anabina. Anabina is a cyanobacteria. And what it does is it basically can do photosynthesis, and it can fix nitrogen at the same time. And so really, Anabina, all it needs is some water and some air and some light, and it can grow a biomass, which is amazing, right? But the way Anabina does it is by actually dividing into different cell types. So this is a picture of Anabina, and you can see that you have these vegetative cells, which are photosynthetic, and you have these heterocysts, which are fixing nitrogen. And if you look at a really long chain of Anabina, you can see that the heterocysts are spaced very nicely apart from each other. So this looks almost like some jewelry. Looks like some nice necklace maybe, or I don't know. And the reason that this happens is that basically these heterocysts make glutamine. They share the glutamine. As the glutamine level drops, there's not enough to sustain life. The next cell here will actually differentiate and become a heterocyst. And this goes on and on and on. But the question is, why do they actually have to do this? Why can't the cells that photosynthesize make their own nitrogen? And the reason is that nitrogenase is a very, very expensive enzyme for the cell to make. And about 70% to 80% of all the protein that this cell makes is nitrogenase. But nitrogenase is very, very sensitive to oxygen radicals. And so you cannot do photosynthesis where you have oxygen radicals and fix nitrogen. So Anabina said, okay, well, I can't do it at the same time in the same cell, but I can still do it at the same time if I differentiate. And I think that this is what we are sort of seeing in other systems. And so that's sort of one example of this. And that is really what I'm most excited about. If we have more time, well, actually, a lot of this work was done in industry by my lab, but my lab here at UNC, we have a rotation student and a lab manager. They're really helping me to get this started. So we're going to do a lot more of this single cell stuff. If there are questions, we can go through questions, or I can show you guys what this looks like in a pathogen. I don't know if you guys are more interested in pathogenesis. I'm happy to do whatever is best for the group.
Yeah, we can do question now, but I understand if I understand you might have an opportunity to share with us some research opportunities, because these are master students and they're looking for thesis projects and something like that. I don't know if you want to share us something in that field, please.
So work that I'm currently working on?
Synthetic biology.
Synthetic biology type work. So there are some synthetic biology work that we're doing. Some of it is very technical. So some of the stuff that I'm working on that is synthetic biology is trying to engineer some enzymes that write RNA to write DNA so that we can make the probes cheaper and better. And so this is a little bit of protein design that my lab does. And trying to understand how basically these communities work, with the idea that at some point you can actually make synthetic communities that will be better. We're not quite making synthetic communities yet, but that's part of the point. And in industry where I came from, so this is not entirely synthetic, although it is related. When you grow cells in giant fermenters, at some point the cells stop producing protein enzyme. So when I worked in DuPont, we had fermenters that were basically gigantic multi-ton bioreactors, silo sized. And in these reactors, you'd have to empty all your media, empty all your cells once every 4 days, because they would stop producing enzyme. And what we see with single cell biology is that basically there are cells that still produce, but most cells don't. And the question is, how do we actually get cells to sense conditions and continue to produce enzymes? And so those are all projects that are biotechnology related that my lab is interested in. We work a little bit more on infection because this is a medical school. And so that's why a lot of the more engineering stuff that the lab does is trying to make these technologies better. Yeah.
I understand that Professor Francisco had 2 questions.
Hi, do you hear me?
Yes, I hear you.
Yeah, thank you. Great talk, first of all. I just had a couple of questions. Do you think this could be like a stochastic activation, like a random activation of different sets of genes that somehow were like selected by evolution as a tool to stay alive in different environments? That's the first question.
Yeah. So I think that's an excellent question about how does this actually how do you give rise to this? And I do think completely that there is got to be some evolved benefit for the cells, right? Some noise is inherent, but why are some programs more noisy than others? And so I do think that there are some things that actually determine that. And so for antibiotic resistance and persistence and tolerance, we are seeing in some organisms that there are genes that allow cells to stay tolerant that are more noisy and come up more than others. I think another part of the question was how much of this is actually a concerted, real physiological state, and how much of it is just some noisy transcription. And I think there's some genes that are differentiated, that are just a little bit of noisy. Maybe a cell needs an extra transcript, it makes it, and we cut the cell right in that second. But what we see with the fluorescent reporters is that some of these really have physiological function. So
it is a fully reproducible, always the same kind of set of genes are activated in the same condition as what I...
When we repeat it, we see the same genes. When we do it with different bacteria, there's some bacteria that have more than others, but there are some processes that are more often heterogeneous. So I mentioned arginine metabolism was more often, but a lot of the attachment apparatuses, pili, flagella, things that have to do with motility and attachment, and secretion, type 3 secretion in multiple pathogens, are really heterogeneous. And there you would think that it's probably because the cells have a strategy when they're infecting, where some cells are going to want to attach and some are going to want to inject with their type 3 secretion. But we do see generally that there are some similarities between different organisms too.
Interesting. So about the arginine operon, is it due to the media you use? Like you use a different media, you get the same activation, or what is the media that reflects more the natural environment of this bacteria?
Yeah, that's an excellent question, because the natural environment, these are soil bacteria, right? So it's really hard to tell what their natural environment is. And I think that's why in general, soil bacteria have more heterogeneity than bacteria that are living, in other environments. We use minimal media, and the reason I use minimal media in as many of these experiments as I can is because that means the bacteria actually have to work. They have to make everything, right? And so we just used M9 media in this particular condition. I don't think that M9 media is similar to soil, because there's no glucose usually in the soil, and there's no, right? But I use it precisely because I want them to have to make as many things as they can. I don't know if it's an interesting question whether if you tried it in many different media, there would be some conditions that really arginine metabolism. Maybe it's media. Maybe if I grew these on in a different minimal media, it would be leucine synthesis. That would be very interesting. I just know that for the typical conditions that I've used, we always see arginine, and I don't know why yet. Yeah.
It's very interesting. I am more from the fungal area. I work with Candida albicans, and in my previous lab, we realized that the amount of sugar we use, the glucose in the media, is really ridiculously high. It doesn't reflect at all the conditions in the body that, as a pathogen, it would encounter. So I don't know. It's just a lot of room to play, I think.
Yes.
So yeah.
It certainly is. And, like people who've looked at heterogeneity in, and I know, different fungi, different and different yeast, but in Saccharomyces cerevisiae, there are times right before glucose shuts down, if there's a second carbon, where you get cells that are switching earlier than others. And so some of these genes might actually be, evolved to sense the sugar conditions and do their deoxy sooner than others. And so you can imagine that, like, it's not just heterogeneous in a static way, but are some deoxys faster than others? What are the rates in which cells switch? What's the heterogeneity in the speed of response? Not just the heterogeneity in yes, no, but the rates that it would be very interesting. Yeah.
Thank you very much. I just have another question. Is it possible? I just had a... Okay, I just remember the story of the Dictyostelium discoideum, the slime mold. in which there is some kind of selfishness embedded in the same species or type that is growing. So in the end, some cells kind of sacrifice themselves to make the stem, and then only a few get to reproduce to make the spores. I don't know if something like that could happen in the bacteria, like some groups would have more, maybe like RNA of some selfish gene that will make them, okay, let's wait. These other cells are going to make the job for us. I don't know.
Yeah. So like that's another one of those group behaviors. So in Bacillus subtilis, there's actually fratricide, where cells will kill their neighbors. So it's not that cells will sacrifice themselves and die, but they'll actually be actively killed. But that takes place in later growth stages. So in the conditions that we use, you don't see it, but it's the same idea that basically cells will either sacrifice or be killed for the good of the community. And we certainly see that. And then the question is, how do you regulate that, and who decides who lives and who dies, and how does how do you fine tune that behavior? Right, you don't have a brain or a calculator, so how do you calculate?
Yeah. Thank you very much. Very, very nice question. A very nice answer, actually. Thank you.
Yes. Thanks, Francisco. More questions? I can show you I mentioned earlier the type 3 secretion in Fimbriae. I can show you a couple slides of that if people want to see, or I will mention that for students who are thinking about this kind of work after your master's, I don't know what the exact procedures. I'm new here at UNC, but UNC is a great program, and so trying to come here, I highly recommend UNC. So applying, or and again, I've only been here for 6 months, so I don't know exactly how all of that works, but we have a very wonderful community here, and Ruben can probably speak to that.
Yeah, that's true. Like, it's a really good program with really nice professors and research. So if anyone is interested, just let me know.
See some stuff in the chat. Should I? It's Francisco. Okay. Should I show some pathogenesis images or?
Yes, please. We have time.
Okay, excellent. So in this work that I did when I was still in industry, we looked at an infection by an organism called Clostridium perfringens. And what's interesting about Clostridium perfringens is it's actually the fastest dividing bacteria, so organism really, other than viruses, I guess, known. It divides in about 6 minutes, and it can cause disease. And it causes disease in humans, but that's not the main problem with perfringens. The main problem is that it causes diseases in chickens and in other birds. And so this is basically the intestine of a healthy chicken. And if you basically have a chicken that is infected with Clostridium perfringens, it has all these lesions and basically gets very sick. And these lesions are caused by a toxin called NETB. NETB basically forms a pore that goes through the epithelial cells and basically destroys them. This is a monomer, and then it makes like a 7-member ring. And so what we did is we basically looked to see whether this organism in single cells is differentially expressed. And so here I'm just showing you UMAP, which is another way to represent these populations. Each dot here is a cell. And instead of showing the heat map, basically they're closer together and colored if they're more similar to each other. And you can see that if we look at where the toxin is expressed, there really is one main cluster that expresses it, maybe a little bit in another, but it's very low expression in clusters 2 and 3. And when we do look at clusters 2 and 3, there are different metabolisms that the cells in these clusters are actually expressing. And so what we set out to do is we said, hey, maybe if we give this bacteria the environment that it likes, maybe we'll make less toxin. And when we did that, we found out that these cells in the other clusters like acetate, if we give more acetate in the total population, the net B level goes down. So there's a high level of net B and then it drops. And even more importantly, in the cluster of cells that are expressing net B, that gene expression goes down. And you can see that the most differentially downregulated gene in this whole entire cluster is net B. And so we were able to actually find out that toxin is only expressed by some cells and find out what the cells that are not expressing like, give them that compound, and then reduce toxin expression. And not only do we reduce toxin expression, the size of the cluster, so this is cluster 0, also goes down. And so we can basically lower the toxin by adding a metabolite that we predicted. And you can see that when we add the metabolite, so this is the media, and then the media with the metabolite, we basically make less of the toxin. So this is one thing that we've done in virulence. We can also look at phages. So if you induce phages in Bacillus subtilis, you can induce them by creating DNA damage. You create DNA damage, you see that there's a defective prophage called PBSX. Some cells express it, and so we don't really know why some and why others. The interesting thing, though, is that Bacillus subtilis has multiple phages. Another phage, SP beta, is expressed in different cells, but you can really see that it's either or. Cells are either in this cluster making PBSX, or they're making SP beta, and we don't know why they're doing that. One thought is that maybe what determines which phage you express is the type of damage. Or maybe it's what state the cell is in to begin with before it's damaged. But what I really like the most is the idea that maybe whichever phage starts first is going to take over, because the first thing that a phage does is it expresses its own machinery and it sucks up all the nutrients and all of the things that are available within the cell. And so the other cells are not able to compete. And so maybe that's what happens. This is in Salmonella. So in Salmonella, basically bacteria can get inside of the cell. And Salmonella can infect different cell types. So it can infect macrophages that engulf it, or it can get into these cells by using a type 3 secretion system. These are like tiny little needles that the bacteria make. And when we tried to study this, we studied it with organ-like 3-dimensional tissue, like an organoid that you can grow. And so we buy these organoids. They have brush borders. They look a little bit like what you would see in the end in your gut. And we basically have a lot of different cell types. They're made out of human stem cells. And we basically take these organoids and we put bacteria in the same orientation that they would be in infection. And what we see is basically that the type 3 secretion, which is what allows these cells to invade, is really expressed by a subpopulation of cells. But fimbriae, which are these pili that allow it to basically enter by a different way, attach and enter in a different way, is expressed by different cells. And I think that the reason for this is a little bit like the reason that I'm interested in arginine, that maybe these processes are incompatible. And what I mean by that is, if you look at these two different processes, they're both things that are on the surface of a cell. So these are your pili, and you can see here microscopically, a pili is about, a micron or half a micron long, and they're all over the cell. These little type 3 secretion, what they do is, this is an electron micrograph of them, but they're tiny. They're just 50 nanometers. They protrude out of the cell, and they have to come in contact with the mammalian, with the host cell's membrane. And so what I think is maybe happening, this is my hypothesis, is that cells that have these really long villi might not make sense for them to have these tiny, tiny, tiny short needles, because the tiny short needles might never get to the membrane, right? It might be precluded by this really thick coat of villi. And so that's some of the work that we're doing with pathogenesis, which usually the story is a little bit more fluid, but I'm happy that I got to show and share. Yeah. There any other questions?
I have a question. Is it
Yeah.
possible? Yeah. I was thinking, could it be possible to engineer Salmonella to produce just a very short fimbria to test if that's true, that what you say?
So that's a really nice idea. I've been thinking a lot about how to test this kind of stuff, and because there is an advantage to having Fimbriae too, right? And so there are in the literature, in the Fimbriae apparatus, one of the components actually determines the average length of the Fimbriae. So I think that you could make them shorter. What I thought would be easier is to just basically lock Fimbriae on, have all the cells make Fimbriae, ensure that they're less able to infect. And if you look at the literature, people have actually shown that if you have Fimbriae, which are usually associated with disease, but if you have them locked on, you have a less effective infection. But that's not as conclusive as showing that you can actually make them shorter. And so, yeah, that would be fun. So that's the kind of stuff that engineering would be really neat. And the question, for synthetic biology, really the question is, how do you engineer cells to go from one state to another in a way that makes sense? And how do you actually get the most out of a population by increasing what it can do? And we're still trying to understand why bacteria do this, but I think that there'll be a lot of really interesting stuff to do once we do. But yeah, I think that's a great idea to make the Fimbriae shorter.
Thanks a lot. Yeah.
I think that will be all. Thank you very much, Adam, for your presentation.
Thanks, everybody. If you're interested in any more information, just please send an email. I think Ruben or Matthew probably have my email address, so feel free.
Yeah, I got it. Thanks, and obviously you will get your certificate about this conference, and let me know if you need anything about from Yachay, please.
Okay, thanks. I think I'm good. This is perfect. It's been a lot of fun meeting you. So
Okay, thanks to everybody, and see you the next session the next week.
Thank you very much. Thank you.
Bye.
bye, everybody.
Bye-bye.